And we are presenting a poster on the phase 2 neoadjuvant trial of the anti-B7H3 antibody Enoblituzumab in men with localized prostate cancer. It was a investigator-initiated trial out of Johns Hopkins in collaboration with Macrogenics, looking at the safety, efficacy, and immune correlates. And this is really the preliminary data being presented for the overall trial, and we hope to put this into publication in the near future. Overall, B7 is a homologue in the B7 superfamily, akin to PDL1, PDL2, PDL3 is B7H3. And we have now got good evidence that B7H3 seems to be highly overexpressed in the prostate tumor microenvironment by prostate cancer compared to even PDL1 and PDL2. And so it's emerging as a contender for what we believe is probably one of the reasons that current immunotherapies haven't really had much of an impact yet in terms of benefit within prostate cancer, and there's no current FDA approvals of any of the CTLA-4 or PD-1 axis agents by the FDA in prostate cancer specifically, possibly because of these physiological reasons. And so our hypothesis was that in the neoadjuvant setting, men treated with who have intermediate or high-risk prostate cancer, as we selected for the demographic, would have reduced biochemical recurrence and potentially good safety and tolerability of the agent, as well as having strong modulation of the local tumor microenvironment that could be seen in this very early phase 2 trial. For our design, we selected men with intermediate to high-grade Gleason scores who wanted to get a prostatectomy, gave them 6 doses or 6 weeks worth of Enoblituzumab, and then they proceeded to prostatectomy. We had a lot of immune correlates from blood samples and pre- and post-treatment tissue that allowed us to really heavily interrogate the impact of this agent on the tumor microenvironment, on, of course, the tolerability clinically to the patient, and, of course, on the clinical outcomes. And we present that overall, tolerability was quite good, with about 12% of patients showing grade 3 or 4 toxicity. Only really 2 immune-related adverse events of grade 3 and above were noted. One patient had what could have been related immune myocarditis that was treatable with steroids, and another had a grade 3 infusion reaction. But overall, again, quite tolerable in the setting of what we know compared to other agents in this space. And then we also saw that patients had PSA responses prior to prostatectomy that we think could be attributable to the drug. And we, of course, present for this single group of patients, a single-arm trial, the Kaplan-Meier data showing time to recurrence post-prostatectomy, with a good number of patients at the 3-year mark having controlled PSA-undetectable disease. And then finally, we were very excited to note that in a promising fashion, the local tumor microenvironment seems to exhibit signs of post-treatment upregulation of CD8 cytotoxic T cells, which are an important signal of potential immunotherapeutic activity. And beyond just the presence of the cells seem to exhibit that the tumor microenvironment had increased granzyme B, which is a measure of activity of cytotoxic T cells. And as well, there seemed to be myeloid lineage cell activation, showing that the myeloid cells were potentially more active as well. Now, there remains a lot of work to still investigate, in which we're in the process of doing, of trying to understand what exactly those tumor microenvironment changes mean, what it means both on the T cell side and on the myeloid side, what the genes that we see upregulated mean in that context, and then finally, which patients, if there's any way to predict which patients, based on their correlative biology, are the ones who responded or had resistance to the agent, and then, of course, how to best optimize the agent for future clinical therapy and in what setting of the disease. That all is ongoing work with combination trials being planned and trying to better understand how this novel potential therapy can be added to the armamentarium of treating prostate cancer, both potentially at earlier stages, as in this case with localized disease, and of course in later metastatic settings. And as we present at this meeting as well, with our ADC, antibody drug conjugate targeting B7H3 or MGC018, it's possible that the metastatic setting also has response to B7H3 targeted agents. So overall, between the two studies, we are highly encouraged that B7H3 is a novel and potentially highly relevant clinical target for cancer, and then especially given our preponderance of data in metastatic castrate-resistant prostate cancer, and now in the neoadjuvant prostate cancer setting, that prostate cancer is a both highly expressing B7H3 cancer, and then also might be therefore amicable to treatment with B7H3 agents. Thank you so much.